Hello everybody, this is our final lecture together and it's about the microglia. So microglia are neuroimmune cells and they have been called their friend or foe, defender, attacker, watchdog, the good, the bad, the ugly, synaptic stripper. These are just some of the names that I could remember offhand. And they have been really acknowledged to be very important in the brain, both development, during adulthood, and then during aging in neurodegenerative diseases. That there was a whole issue of science devoted to microglia a few years ago, and here this beautiful purplish microglia is shown how it interacts with the neurons, with these fine processes. Microglia are one of the glial cells in the brain, and I just want to start, in case you just haven't been aware of this before, that glial cells constitute half of the human brain. This is different across evolution. I just want to make sure. In mice, they are less than half of the brain cells. But in the human brain, we have around 86 billion neurons, and then there's around 84 non-neuronal cells. The neuron to glia ratio differs depending on which brain region you're talking about. In the cortex, it's much higher than, for example, than in the cerebellum, in which you have 0.23 non-neuronal or glial cells to neurons. And when I say glia here, I actually include, or the researchers who did this work include both astrocytes, microglia and oligodendrocytes. In principle, they just made the soup of the brain and then labeled it with neuronal nuclei, antibody, and then they just counted how many neuronal and non-neuronal cells were in different brain regions. So this is important because, again, even though the CNS has been studied in large part by looking at neuronal function and being defined by neuronal function, it is important to know that the 50% of the cells in the brain are actually glial cells, and they can do many things. So Eric has talked about astrocytes. Today I will be talking about microglia. Microglia are pretty mysterious, one of the most mysterious maybe at this point, except maybe oligodendrocyte progenitor cells, brain cell types. They're usually called brain resident macrophages or innate immune cells of the brain. They are really the interface between immune and nervous system. And again, this is pretty interesting for me because each of these systems, immune and nervous, are really crucial for our perception of the external environment. Microglia are known to support and modulate neuronal activity. Here represented by their different motility that is actually influenced by neurons, and also remember those beautiful processes in the first image that I showed you that are in touch with neurons. They are main contributors to the development. We know that during development it can lead to removal of extra synapses, but also it can phagocytose the extra neurons that are going to be dying. They are really critical in maintaining CNS homeostasis, and we know they play a very important role in aging and then in neurodegenerative diseases or any brain insults. Now, what are the microglia? What is their origin? They are really different from the rest of the brain cells in their origin because they are not. Their origin is not from neuroepithelium as other brain cells are. So normally in brain parenchyma you have neural progenitors that during most of the early neurogenesis is going to make neurons, and then during late neurogenesis is going to make neuron astrocytes and oligos. On the other hand, microglia, we now know that they are derived from the primitive myeloid progenitors that are derived from the yolk sac, and that they enter the neural tube at the early embryonic stages around embryonic day 9 in rodents. So they enter the neural tube before there is a blood-brain barrier formation, so they can freely enter it. This is actually pretty interesting and I think quite important and not very well studied, because that means that during these early stages of embryogenesis where we have active division of neural progenitors making neurons, actually the microglia are pretty much the only main glial population during the large part of this. This is illustrating this with the addition of blood-brain barrier. Just to illustrate again, these are yolk sac progenitors enter into the or colonize neuroepithelium before there is a blood-brain barrier formation. Macrophages in the other tissue are replaced. They also are made from these progenitors. In the other tissue, they are during fetal development replaced by fetal liver monocytes, and then during the adulthood, they're replaced by bone marrow monocytes. In contrast, microglia actually are self-renewing in the brain throughout our life, and there is an evidence actually that during the damage on insult, when you have disease, in the case when there is a blood-brain barrier breakage, some of these monocytes or macrophages actually can infiltrate the brain. But under the normal conditions, microglia in the brain will all going to be derived from these early colonizers, which indicates that actually they have a memory of our early embryonic stages until the adulthood stored in themselves. My apology. MAFB, which was one of the transcriptional factors that characterized adult microglia, if you perturb that gene, you end up in inflamed microglia and a chronic inflammation in the brain. So this would be genetic perturbation that researchers have done, but those can also happen in people who inherit different mutations in these genes. Or if you have some environmental perturbations of these pathways, they can disrupt these stages, and that can be associated with neurodevelopmental disorders, even if that neurodevelopmental disorder may not be actually manifested until you age to certain come to a certain age, and then you're more sensitive to development of some diseases. In order to develop microglia, there's a crosstalk of microglia in the CNS cells. So as I said, after they colonize and before, they will actually end up tiling the whole brain, and we'll talk a little bit more about tiling of the brain mean. And they again acquire different morphology, but they also again have this different gene expression. And this differentiation is influenced by signals from the maturing CNS, so from neurons and from the astrocytes. They influence this microglial development, and those signals can be CSF1, colony stimulating factor 1, which is really important for their development, IL-34, TGF-beta, so they all play a role in microglia development. Again, expression of core genes during development, cell of 1 is really crucial. In addition to these, we know that sexual identity plays a really important role in development of microglia. So microglia are different between males and females, not only by gene expression, but also by density in certain brain regions. We also know that systemic signals such as microbiota or any inflammation in utero in mothers will be able to affect microglia development, and also local neural cues do influence microglia development. And it's actually thought that this last part for local neural cues influencing development that lead to this really fascinating microglia diversity that we see in adult brain. So the study, the figure, this histogram or dot plot on your left shows you different brain regions for frontal cortex, occipital cortex, hippocampus, putamen, and cerebellum. And this pretty much shows the cerebellum is mostly different than all the other regions. And if we have time, we can discuss why it would be cerebellum. But also that, these brain regions mostly, you can identify several different brain regions and then signaling pathways that are mostly exhibited in these regions. For example, in cerebral cortex, it's immunoregulation. In corpus callosum and cerebellum, we have phagocytosis and lysosomal activity. In hippocampus cerebellum, we know that there are genes that are involved in energy metabolism in the adulthood. So again, these different microglia in different brain regions, the whole point of this slide is to tell you that microglia in different brain regions have differential gene expression. So they're diverse. When we say microglia, it doesn't mean exactly the same thing when we talk about the cortex as it does mean when we talk about hippocampus or the cerebellum. This, of course, will have implication not only for neuronal function in health, but maybe even more importantly, it can play important implication in disease conditions. For example, if cerebellar astrocytes have a higher energy metabolism, or if they have higher phagocytosis, they may be more prone to induce neuronal death through this phagoptosis, or a mechanism by which microglia may be able to induce neuronal death. This slide is just going back to what I said. So this was showing how neuronal local clues can influence the neuronal microglia heterogeneity. Now I'm just going to show you some evidence about RNA sequencing that highlighted the sex differences in adult microglia. So if you do the staining of the brain, and as I said, you can see that there is different in microglia density in different brain regions, and that can also be affected by different drugs. But when these newer methods such as RNA sequencing were done, so you would isolate microglia from either male or female brains, and then do RNA sequencing, and then differential gene expression, and then pathway analysis. You can see that there are a number of genes that are different in females versus males. So these are the genes that are more expressed in male microglia. So these are just microglia isolated from the brain. And these are the genes that are highly expressed or enriched in female microglia. And then when you look at the pathways, these are the pathways that come. So for males, the first pathway that's different is inflammatory response, connected with transcriptional factor NF-kappa B, which is like the star molecule of inflammation. On the other hand, when you look at the females, you mostly have neuroprotective pathways that seems to be enriched, which led to this schematic. Then when they use these luciferase mice, so these mice, you have luciferase gene, which makes the light. It's firefly luciferase that makes firefly glow in the light, but you have a mice in which you can express in under NF-kappa B regulation, and then you can in vivo look where NF-kappa B is activated. So these are again mice. You see that it's really significantly activated in the head. So when you compare NF-kappa B luciferase between males and females in the whole body, there isn't a huge difference, or there is no significant difference. But when you isolate microglia from males and females, you can see that there's almost double activity of NF-kappa B in male microglia than there is in females, again confirming these RNA sequencing results. This, of course, can play an important role in development of diseases in which we know that male or females are more affected, and we can discuss this further. Now, what are the microglial functions during development, both pre and postnatal? We know that they can both induce cell death, and here is an example of this, and both embryonic stages and also early postnatal stages. So they can induce it either by secreting pro-apoptotic factors, even NGF, which is thought to be its neurotropic factor, but at that time these progenitors are expressing the receptor B75 that actually leads to apoptosis. On the other hand, there's evidence that it's important for neuronal survival and survival of oligodendrocytes. And again, this is shown in postnatal deals, postnatal day 8 and 20 for oligos, and postnatal day 5 for layer 5 cortical neurons. They also are important for axonal outgrowth, so they can regulate axonal outgrowth, and they're also important for fasciculation. They help with cortical neuron migration to different layers, and very importantly, they play a role in synaptic development. So they can both promote the spine formation, so there is evidence in these different early postnatal days, but they also lead to presynaptic pruning. And again, we know that this is actually regulated together with astrocytes. We know that astrocytes that are in contact with these neurons, they are secreting IL-33, and that leads to the activation of this presynaptic pruning by microglia, which is a beautiful example of how two different glial types, astrocytes and microglia, work together in this case in a normal developmental process. This again, pruning of these synapses is really important because again you want to this is one of the hypotheses how this happens. So there's this recruitment signal which is most likely this IL-33 that we just discussed is made by astrocytes. But in addition to that, it's thought that there are some eat me signals on the synapses that are less used or less active intact. Synapses, on the other hand, these more active synapses do not express those signals, so microglia come and remove the synapses that are silent or much less active. We know that some of these signals are complement receptor because there's a beautiful work showing that complement receptor knockout in microglia do not remove some of these synapses in different brain regions. In adult brain, we know that microglia have the processes are highly motile. They constantly survey their niche, so they sense different molecules produced by astrocytes and neurons as well. So they listen to neuronal activity, expressing these receptors on them, but they can also sense different chemokines, scavenger receptors, purinergic receptors to tell them about injury. They're involved in the clearing of apoptotic neurons, and neuronal progenitors in adulthood, and again they keep being involved in synaptic pruning, and this overt synaptic stripping is thought to be one of the major mechanisms that we'll talk how they can contribute to neurodegenerative diseases. This is what I just mentioned briefly before, that there is a tiling of microglia in the brain. So there was a beautiful study that looked how much both astrocytes and microglia overlap, neighboring overlap in the brain. So for microglia we know, so again this is how far they go, and this zone here, the Z, is the part that doesn't show overlap. And in astrocytes, while they're mostly tiling the brain, in different brain regions we have more or less of the overlap. In microglia, so there is no overlap in the healthy brain, and it's thought that they called it physical independence, and they think it's an optimal strategy to maximize this efficiency and specificity with which microglia can interrogate the space around them, so their microenvironment, because each one of them, if they have a separate, then they can really react just to that changes that they can collect. So what happens when they sense something? What I want first, the problem for you to think about is to see how they're actively surveying neuronal activity. So what you're using microscopy. So you have your microglia labeled with EGFP. So it's expressed under microglia specific promoter. So it's either IBAV1 or CX3CR1 or CSF-R1. Any of those might actually exist. So they express GFP under that promoter. So you can do confocal in live mice. And you want to look whether neuronal activity affects mobility. So you're measuring how quickly are these processes moving. So you have either velocity, or you can determine this, how much space they cover, either way. But how would you test whether neuronal activity affects this mobility of microglia processes? I want you to think about that. So I just want to briefly spend some time before we go further about roles about astrocytes and microglia and neuron communication. This slide is too much. I really don't expect you to memorize nothing here, except that both astrocytes, microglia, and neuron may be forming that quadripartite synapse. You know how they thought about astrocytes together with presynaptic and postsynaptic neuron forming the tripartite synapse. I think we should probably revise it because we know that microglia are sensing neuronal activity, and again, hopefully with your think about your problem, you have come up with the experiment that could test that, whether the process are more. But these are all the factors. So in blue are the ones that are secreted by or produced by microglia. Beige are the ones from astrocytes. All of the purple ones can be produced by multiple cell types, and the red are the ones that are produced by neurons. And again, from presynaptic neurons, and these green are from postsynaptic. And you can see what receptors, what do they bind on microglia, on astrocytes. So this shows you that there's so many molecules by which microglia can influence astrocytes. And again, we will go through a problem showing how microglia actually can influence astrocytic or astrogliosis in disease. But we also know that astrocytes again can play important roles in pro-inflammation of microglia. And again, producing a lot of these neurotropic factors for the neurons, so they are both very supportive. Now I want to spend some time what happens with microglia with aging. This is important because, again, this is neuroimmune axis, and we know that lots of neurodegenerative diseases are associated with neuroinflammation. And again, neuroinflammation seems to be associated with aging. So this was a study from humans, actually, in which they did analysis of samples from humans across different ages and then divided them. Here is their age. And did a gene expression of microglia genes, oligodendrocyte, astrocytes, and neuronal genes. And they have actually found very strong correlation of microglia gene expression and astrocytic gene expression, so glial genes, not as much, with the age of people. So they're really good predicted. They were really good at predicting the age. Microglia were really good predicting the age of the humans based on their gene expression. So, which indicates that microglia gene expression changes with aging. What we know both from mice and from humans, that when they age, microglia seem to become more immune primed. So they seem to be expressing more inflammatory markers, and more so here MHC2, so these are expressed antigens. CD68 is involved in phagocytosis. Toll receptors are recognizing different pathogen-associated receptors, such as LPS, for example, which is bacterial lipopolysaccharide. And then integrins also play a very important role in microglia activation. Then we have cytokines, such as IL-6 and IL-1 beta. They are both associated with many diseases. For example, we know that in Huntington disease, patients have increased systemic IL-6 levels many years before they actually start showing the symptoms. In addition to these gene expression changes that again indicate more activated microglia, they do change in their morphology, and they lose some of these processes. And you can think if they're losing some of these processes, their ability to sense neuronal activity or their environment may also be affected by this. What implications does it have? Well, the simplest neurobehavioral complications that can be associated with this increased reactivity or priming of age microglia are just when your simple viral or bacterial infection that you had. We know that when you have viral or bacterial infection, when you feel sick, there is CNS response. It's usually induced by cytokines that go into the brain. Small amount of them go into the brain from periphery, and then they lead to microglial activation, which then does produce cytokines, which will usually lead, you would feel, that's why you feel like you want to sleep more, you have a little bit of lethargy, social withdrawal, reduced appetite, and all of these actually are very beneficial because the way evolutionarily we were developed to fight these infections is by raising the temperature or by fever. And then if you move less, if you're lethargic and sleep more, you're gonna maintain this high increase in fever and defeat the pathogen. Social withdrawal, it's probably also beneficial response because again, you are trying not to transmit whatever infection you may be having. It's a way of social distancing that our brain has developed. Now, if this happens in the age brain, so this is what happens with you guys if it happens to me, it may just amplify and induce all of these other, already, now that these behaviors that were here really good, they may actually become really bad to the older people. So they become maladaptive. In addition to that, it is thought that this primed state of aging microglia can influence onset, severity, and progression of neurodegeneration. Here's shown example for Alzheimer's disease and Parkinson's disease. So, for example, in Alzheimer's disease, these primed microglia may be less efficient in phagocytosing beta. They may actually lead to induce, so these are functional impairments, but in addition they can actually then phagocytose more, remove more synapses that should probably be removed in that day. So this shows us how this aging of the microglia, in addition to having these consequences with the regular diseases, they can help towards injury. So is that why microglia are going there? We know that they do express ADP receptors. Okay, so microglia activation of microgliosis, it's similar to astrogliosis. It's a process of activation that includes morphological, gene expression, and functional changes. Morphological changes can go they're from the human brain. They can become hyperamified, they call them bushy, so they have thickening of the process, or they can become amoeboid microglia. We also know that this is the study that look at microglia, single-cell RNA sequence microglia in Alzheimer's disease, showing that they actually have two disease-induced transitions from stage 1 disease-associated microglia that may, or stage 2 associated. So they have two distinct populations of microglia. It is not really clear. They're shown here as red and green, trying to indicate that they might be having a neurotoxic versus neuroprotective effect, but that still has to be proven. But there seems to be two separate, and there's, I think, much less of these. And TREM2 is necessary for this transition. Knowing that TREM2 is one of genetic risk factors for Alzheimer's disease, I think that was the reason why it may have been thought that they would be protective. For Alzheimer's disease, there were several studies that have indicated how in neurodegenerative diseases, microglia may be affecting CNS. So in addition to what I have described, in any injury in which there might be, again, these prime microglia may be secreting more pro-inflammatory cytokines, which can then negatively impact, they can be neurotoxic for neurons. This synaptic stripping, so they normally strip synapses in development and in adulthood when need be, but it seems that that is actually exaggerated in Alzheimer's disease. And here, three proposed mechanisms. One could be that there is this hyperactivity in neurons, or tau oligomers, or defective mitochondrial neurons may lead to the expression of complement 3, and then which is recognized by complement 3 receptor microglia and being cleared. Additional thought that these complement factors C1q and C3 and C3 receptor may be there because there is this function, there is just these synapses are actually targeted by some other way, and then they come and they remove them. We do know that, for example, and we will talk about that problem, we know that astrocytes are known, for example, to secrete in Alzheimer's disease complement 3, and that could be the way, for example, in addition to these shown here. Third pathway they propose that there might be phosphatidylserine shown in neurons. So phosphatidylserine should normally just be on the inside of the neurons. When it's on the outside, it is an eat-me signal for microglia, and that's usually shown in apoptotic cells. So idea is here that the dysfunctional synapses will be showing this phosphatidylserine on the outside, which will then be recognized by TREM2 on microglia, which will then lead to the engulfment of these synapses, or if it is on the dysfunctional neurons. So that will be all that we will be covering the class. Again, very brief, going through to microglia development, trying to learn where they come from, what are their origins, that there are different stages of their development, and then microglia are really sensitive to different factors during their development that include both sexual identity, again, infections, systemic responses to infection, and also neural cues that they end up having difference in adulthood with different brain regions. So microglia are not uniform across the brain, they're heterogeneous, and that they have different functions that may be playing more important role in these brain regions, that they show sexual differences between male and female, that can then contribute or prevent different diseases with aging. We know that with aging, microglia change. They seem to become more primed, and they also have some loss of function, it seems, in sensum. We know that some of those changes, especially those that lead to more reactive phenotype, can also contribute to the onset of neurodegenerative diseases. And we have just went quickly over how in the Alzheimer's disease, this extra remover of synapses by microglia can actually promote synaptic loss, and then all the cognitive and behavioral changes that come with a reduction of synapses in Alzheimer's disease. Okay, bye.